Khatib series in Human Anatomy and Physiology. Chapter 2. Cell Structure and Function. Episode 10. The Cell Cycle and its Regulation. If you enjoyed the video, don't forget to hit the like button and share it, to help us reach every medical and health professional student around the world. And of course, make sure to subscribe to the channel for more valuable content. Your comments are highly appreciated. Welcome to today's lecture on one of the most essential and tightly regulated processes in biology, the cell cycle. The cell cycle refers to a sequential and highly orchestrated series of events that a cell undergoes in order to grow, replicate its DNA, and ultimately divide into daughter cells. This cycle is fundamental to both somatic and reproductive cells, although the outcome differs. In somatic cells, the cycle ends with mitosis to produce two genetically identical cells. In reproductive cells, the cell cycle gives rise to gametes through a specialized process known as meiosis. Regardless of cell type, the cycle is composed of distinct and well-regulated phases, G1, S, G2, M, and occasionally G0. Let's begin with the G1 phase, or also known as gap1 phase. During this stage, the cell undergoes active growth and metabolic activity. It increases in size, synthesizes RNA, and produces essential proteins. Also, the cell duplicates its organelles such as mitochondria, ribosomes, Golgi apparatus, endoplasmic reticulum during this phase. This ensures it has adequate nutrients and energy to support the next phase, which is DNA replication. Therefore, G1 phase of the cell cycle is a very critical and essentially a preparatory stage, where the cell gears up for the journey ahead. Following G1 phase is the synthesis phase, or S phase. This is the stage where DNA replication occurs. As shown in this image, each chromosome is duplicated, ensuring that after division each daughter cell inherits an identical set of genetic material. Additionally, histones and other structural proteins are synthesized to package the newly replicated DNA. Next to this phase is the G2 phase, or gap2 phase. This stage is the final stage before cell division. Here, the cell continues to grow and ensures there's enough volume, cellular components, and organelles to distribute between daughter cells. During this phase, the cell also synthesizes the proteins essential for mitosis. These include tubulin needed for spindle formation, chromatin condensing proteins, and mitotic regulatory proteins such as cyclins. G2 phase also serves as the cell's final quality control checkpoint before entering division. Herein, the cell performs a thorough DNA damage check to confirm that replication in the S phase was accurate and complete. Any detected errors during this stage are either repaired or trigger cell cycle arrest. Additionally, centrosomes, which are microtubule organizing centers, finish duplicating and maturation during this phase and begin migrating to opposite poles of the cell, preparing for mitotic spindle formation. G1 phase, S phase, and G2 phase are known as interphase and is the longest phase of the cell cycle. After G2 phase is the last phase of the cell cycle, which is known as M phase, or mitotic phase. In somatic cells, which represents the majority of our cells, this phase involves mitosis. Recall from our previous lecture that mitosis produces two identical cells which have the same number of chromosomes as the mother cell. This process is subdivided into four major phases known as prophase, metaphase, anaphase, and telophase. It also includes a final process that divides of the cytoplasm to produce the two daughter cells, which is known as cytokinesis. However, in reproductive cells, the M phase is modified into meiosis, a specialized division that produces genetically diverse haploid gametes, sperm or egg cells. We will cover the detailed steps of mitosis and meiosis in the next coming lecture of this series. Notably, some cells, however, do not continually cycle. These cells exit the cycle into a phase called G0, also known as the quiescent or the resting phase. In G0, the cell remains metabolically active but ceases to divide. Certain cell types, such as neurons, may remain in G0 permanently, while others, like liver cells, may re-enter the cycle if stimulated by external signals. Now we will move forward to cover the regulation of cell cycle. Herein, it's crucial to mention that the cells progress through the cell cycle, they do not move blindly from one phase to the next. Instead, this journey is tightly regulated by a series of surveillance mechanisms known as cell cycle checkpoints. These checkpoints serve as internal control systems that monitor the cellular environment, DNA integrity, and readiness for division. Their function is essential, not only to ensure that each stage is completed accurately, but also to protect the organism from errors that could lead to genomic instability, mutations, or diseases such as cancer. There are three principal checkpoints in the cell cycle, each positioned at a strategic transition point. The first is the G1 checkpoint, also known as the restriction point. This checkpoint occurs near the end of the G1 phase. At this stage, the cell assesses whether conditions are favorable for DNA replication. It evaluates cell size, nutrient availability, growth signals, and most importantly, the integrity of the DNA. If any of these parameters are suboptimal, the cell will pause or exit into the G0 phase, a quiescent state where it can remain until conditions improve. The second is the G2 checkpoint, located at the end of the G2 phase, just before mitosis begins. This checkpoint verifies that DNA replication has been completed successfully and checks for any damage or replication errors. If abnormalities are detected, the cycle is halted, allowing the cell time to repair the DNA. If repair is not possible, the cell may undergo apoptosis to prevent the transmission of faulty genetic material. Finally, the third checkpoint is the M checkpoint or the spindle assembly checkpoint. This checkpoint occurs during the metaphase stage of mitosis. It ensures that all chromosomes are aligned correctly in at the metaphase plate and properly attached to mitotic spindles. Now let's move forward to understand the role of cyclins in regulating cell cycle. Generally speaking, cyclins are essential regulatory proteins that drive the progression of cells through the different phases of the cell cycle. They function by binding a group of enzymes called cyclin-dependent kinases to form a cyclin-CDK complex. However, this complex remains inactive and is activated by another enzyme called CDK-activating kinases. Now the activated complex can phosphorylate specific target proteins, which in turn activate or inhibit processes required for each phase of the cycle. During the G1 phase, cyclin D binds to CDK4/6, promoting cell growth and preparation for DNA synthesis. As the cell progresses toward the S phase, cyclin E activates CDK2, pushing the cell through the G1 checkpoint and initiating DNA replication. In the S phase, cyclin A associates with CDK2 to ensure DNA is replicated accurately and completely. As the cell enters G2, cyclin A binds to CDK1 to promote preparation for mitosis. Finally, cyclin B partners with CDK1 in the M phase to initiate and regulate mitotic events such as chromosome condensation and spindle formation. Importantly, and as shown in this image, the levels of cyclins rise and fall in a regulated cycle, and their timely destruction ensures that the cell does not re-enter a phase prematurely. This precise control prevents genomic instability, uncontrolled division, and tumor formation.